Well, thank you all for coming to this monthly lecture series I'm trying to host, or am hosting here at the university about psychedelic neuroscience. Welcome. So this is the second lecture that we're having. I'm trying to have these every month. So my name is Jessica Nielson. I'm an assistant professor here at the university. I am jointly appointed in the Department of Psychiatry and Behavioral Sciences, as well as the Institute for Health Informatics. And I'm also the founder of the Psychedelic Society of Minneapolis. And so the Psychedelic Society of Minneapolis is a group of psychonauts that have all kind of gathered together to try and destigmatize the conversations about psychedelics and their therapeutic potential and their spiritual and recreational uses, and really trying to get together and give people a safe space to talk about this. We're not an organization where people can come and solicit drugs or sell drugs, but we do try to offer education and advice and resources to people to promote harm reduction and to give people more information about how to engage with psychedelics responsibly. Because they are a pretty hot topic right now, and so I just want to keep everybody informed. And I started this separate psychedelic neuroscience group because the Psychedelic Society of Minneapolis is a private group, and we actually have pretty strict criteria about who comes into the group. We actually have been getting a lot of kind of drug dealers and drug seekers trying to come in, and that's really not the point of the group. But I do also feel a bit of an obligation to put out some education about how to engage with psychedelics responsibly, because they are kind of plastered all over the media. There's lots of popular press about their potential to treat various major mental illnesses. So I still want to be able to get the information out there to the public, even if, you know, everyone can't get into our private community and come to our different events. So this is going to be the second version of that. Still kind of thinking about the topic for next month, so if anybody has any ideas about what they would like to hear about, feel free to let me know after the talk. Okay, so just a disclaimer. So the information presented here is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment recommendations. The focus of this talk isn't really on therapy, but I am kind of touching on a few kind of therapeutic mechanisms that potentially might influence people to think that they might be helpful. So I just want to let you know this isn't sort of professional medical advice by any means. So all this content, including text, graphics, images, and information contained on or available through this talk, is for educational purposes only. If you are in need of clinical help or medical support, please contact your physician. You'd be surprised at how open-minded some physicians are. We do have a lot of clinicians in our community that are trying to create safe spaces for their patients to come and talk about psychedelics and ask questions. So, you know, if you are searching for therapy or some other option, you know, you can talk to your therapist. If you don't feel comfortable with that, we do have a list of therapy providers on our Psychedelic Society website, psychedelicmpls.com. So these are therapists that are open-minded to their clients using psychedelics, but will not give you psychedelics, but can actually just offer you kind of advice on how to maybe integrate your experience or how to approach them responsibly, but they won't be a source for you. Additionally, if you're wanting to get involved in clinical trials, there are quite a lot of clinical trials happening around the United States where people are getting psychedelics and other Schedule I drugs in kind of a scientific setting. Unfortunately, none are happening right at this moment in Minnesota. The closest site is Madison, Wisconsin, and I think there's another one in Chicago. Unfortunately, you kind of have to live there to be eligible for this study. But I'm hoping to change that and get some studies going up and running. But right now, if you want to kind of look into what's available, again, go to our website, psychedelicmpls.com, and then there's a tab for resources in clinical trials. If you are interested in clinical trials, just know that clinical trials are not intended to be a treatment for any of these disorders that they're studying. They're actually just science experiments trying to determine whether there is any potential or risks for an emerging therapy for a given condition. And being in a clinical trial is very time-consuming and intense, and it's not like just fun taking drugs and tripping in a doctor's office. You know, it's a lot of work, and there's some pretty strict inclusion/exclusion criteria. So give it a good thought. You know, if you're considering it, but know that it's really not a place for you to get free psychedelic drugs with a doctor. There are other options available, mostly outside of the United States, if you're looking for access to legal options. Things like, you know, ayahuasca retreats down in Peru or Costa Rica. Psilocybin is decriminalized in several places now, so it's decriminalized in Jamaica, and there's a retreat center down there that offers psychedelic therapy. I don't know how many people are aware of kind of the decriminalization efforts happening around the nation. So Denver actually decriminalized psilocybin a couple months ago, followed closely by Oakland in California, where they actually decriminalized all psychedelic plants. So that includes mushrooms and plants that you can use to make ayahuasca and iboga and mescaline containing cacti. And so that doesn't mean that they're legal, but they are they have this language in their resolution that they passed that basically says people can cultivate, harvest, consume. I don't know if there's a distribution clause in there, but yeah, I guess not. But still kind of an option if you want to go there. I don't know that you can go there. There's going to be mushroom stores like there are in Amsterdam, but we're kind of, I think getting to a point where, the conversations and the stigma around this is starting to loosen up like it did for marijuana, a couple decades ago. So I'm really optimistic that hopefully we'll kind of see a wave and a groundswell of support and decriminalization across the nation. Fingers crossed. Hopefully here too. And also another big disclaimer, I'm a neuroscientist. I'm not a clinician or a medical doctor. I'm also not a pharmacist. So this talk was a little challenging for me to put together. It was kind of a learning experience for me because I actually never even took a class on pharmacology as a neuroscientist. You kind of just learn the basics about different cell types and brain regions and physiology, but pharmacology is a very specific subset that's more rooted in like chemistry. And so just take that knowledge and take that caveat as I present this, that I'm not an expert in pharmacology, so hopefully you can bear with me with some of this content. This is the first time I'm ever giving this lecture. I'm actually trying to pilot content for a course I want to officially have at the university where I teach psychedelic neuroscience, and so it takes about a year to develop a new class, and so I really want to start getting these lectures together, pilot the content with the community, and really refine this. So again, if it's a little rough around the edges, just bear with me. Okay, so first off, are there any kind of questions before I get started? Okay. So just an overview of what I'm going to talk about today. So first, just kind of give you, for those of you that don't know me, who I am and how I got involved in psychedelic research. Next, for those of you that aren't even familiar with what pharmacology is, I'm just going to give you a brief overview of what pharmacology is and why it's important to know this in order to understand how psychedelics work. And then also, what is the current state in the scientific literature about what we know about the pharmacology of psychedelics, and where are there potential knowledge gaps for new discoveries that we could potentially do in terms of research here at the university. All right, so first, who am I? I've been a psychedelic researcher for about 10 years, kind of casually. So when I was in graduate school, I was studying neurobiology, kind of the neurobiology of trauma. And I regularly attended this festival called Burning Man. Who here has heard of Burning Man? Yeah, okay, so I go there a lot, and Burning Man's a fun party. There's lots of cool stuff, lots of drugs, lots of crazy hedonistic stuff, but there's also a lot of like really smart people that are like changing the world, that go there to have fun and also kind of share their skills and their gifts with everybody. And so one of those kind of groups of smart people is MAPS. So this is the Multidisciplinary Association for Psychedelic Studies. Who here has heard of them? Okay, good, a few of you. MAPS is a nonprofit organization that was started by Rick Doblin in the early 80s, right after MDMA was made illegal. So a lot of psychotherapists in the 80s were using MDMA as an adjunct to psychotherapy. And then when the rave scene got a hold of it, and there really started to be a lot of widespread abuse in the public, the DEA shut that down and made it illegal. And that was really upsetting for all the therapists at the time, because they really saw this as a very promising avenue for potential therapeutic adjunct to psychotherapy that now was illegal, and it made all of them criminals trying to treat their patients. So Rick Doblin was like, I'm not having this. I'm going to start a nonprofit organization, and I'm going to harass the DEA every chance I get until we make MDMA legal again. And fast forward 30 years later, they're now in phase 3 clinical trials to treat post-traumatic stress disorder with MDMA as an adjunct to psychotherapy. But that's 30 years and millions and millions and millions of dollars later just to reschedule something that was already being used. So they're basically spending all that time and money to get back to where they were in the 80s, just to convince the government that this does have profound therapeutic potential. So good on him for doing that, but it's been kind of an uphill battle. But I was able to kind of witness what was going on at Burning Man when they were kind of talking about this. And there was also some people from Johns Hopkins there, so Roland Griffiths and Matt Johnson and some other people from UCLA that were doing psilocybin research. at prominent universities like Johns Hopkins and UCLA and Harvard. And I was a graduate student at the time, and I thought, wow, I can't believe this is even an option to do this professionally. I could be a psychedelic scientist. That's amazing. So I finished my PhD, and by the time I finished my PhD, I was actually kind of sick of science, and I didn't really want to do it. I won't go into the details of some personal issues that I had, but I was kind of just totally disinterested in the whole process, and just was thinking maybe I'll just go be a waitress or something. I didn't really want to do science anymore. But through just sort of navigating, I lived in my car for 4 months and was just kind of letting synchronicity guide me to try and figure out what I wanted to do with my life after that, having spent all this time in school to get a doctorate and then not having any interest in using it. And somebody approached me and they said, there's this healing center down in Peru that's giving people ayahuasca to treat trauma. And if you can't afford it, there's this really rich guy that's offering to pay people to go that otherwise wouldn't be able to afford it but could really benefit from it. And so I submitted this video testimonial to this guy, kind of basically claiming my need for this. And I was awarded, actually they called me on my birthday. I don't think they knew it was my birthday, but they said, hey, we're going to pay for you to go to Peru for 2 weeks. All expenses paid. So I was like, wow, thank you. So I went down there, and just serendipitously, I happened to be down there with a group of war veterans that had PTSD, because the guy that started the center was a Marine, and so he had a soft spot, obviously, for military personnel and veterans, and helping them treat their PTSD. So it was like me and 10 guys, basically 10 veterans down there, and I was a little nervous when I first got down there, kind of being by myself as a woman. And but kind of seeing all of their transformation and seeing them soften up and seeing the healing process unfold with ayahuasca was very inspiring to me. And ayahuasca helped me myself, but really what I took away from it was kind of wanting to work with ayahuasca in some sense and working with ayahuasca and trauma. So I came back to the United States and contacted Rick Doblin at MAPS and was like, I want to be a psychedelic researcher. How do I even do this? I'm really interested in studying ayahuasca for PTSD. And they just happened to get contacted by National Geographic and wanted to do a documentary about psychedelics as therapy for the show called Drugs, Inc. So Drugs, Inc. is this documentary where they actually really demonize a lot of drug use. They demonize the dealers and the users and the suppliers and kind of the whole chain of people that are involved in drug use and abuse. But the episode about hallucinogens was very positive. It was very much focused on the therapeutic potential, and that I thought was like a really good sign that maybe the tides were turning, and I felt really good about being a part of that. And then it just kind of started this collaboration with MAPS and trying to actually get a study started up looking at ayahuasca for PTSD, and that was in 2011 that we initially started having those conversations. And so first I actually was just trying to figure out how to get integrated in this community. I didn't have any experience in psychedelic research. I wasn't a psychologist. I didn't know anything about plant medicine. I was just a neuroscientist. So not just a neuroscientist, but just didn't have the right expertise, at least according to what everyone else was doing. So first was like, I had this idea, I had this hypothesis about ayahuasca treating PTSD. So I started giving talks at various psychedelic conferences around the Bay Area. So the first one was this entheogenic symposium that was specifically focused on women doing psychedelic research in the Bay Area. This was hosted by ERIE, which is it's a nonprofit organization that stands for Entheogenic Research, Integration, and Education. They've now sort of merged into this decriminalized nature group that is actually the ones doing all the kind of the work on the ground, getting psychedelic plants decriminalized. So they're a really great organization. I was able to speak at MAPS conference, Psychedelic Science, in 2013. There was a Horizons conference in 2016. Again, just basically doing the rounds, speaking at all these different scientific conferences, getting the word out. I actually was able to do a survey study to study ayahuasca for PTSD, but I was never actually able to get like a prospective clinical study with ayahuasca for PTSD in the United States. It was just very complicated working with the FDA, trying to get this kind of admixture of these two plants that you boil together, and FDA wants everything to be standardized, and it's like they want this pharmaceutical grade version of ayahuasca. And if anyone has taken ayahuasca, it's sort of just like it's up to the person making it. It has these two main plants in it that give you the dimethyltryptamine and the enzyme that inhibits it, so it can be orally active. But then there's other kind of admixtures that they add to it, and I don't know, it started to not sit well with me, this idea of like taking the spirit out of this plant and trying to pharmaceuticalize it and westernize it. And I'm not really a superstitious person, but like ayahuasca is kind of interesting. I don't know how many people are familiar with ayahuasca. taken ayahuasca. So people kind of say that ayahuasca, she like calls you to work with her in some form, but she's very finicky about how you work with her. She has all these restrictions, like you can't eat certain foods and you can't engage in sexual activity, you can't have salt, or she doesn't like other drugs, she doesn't like competing with other things, if you believe in that philosophy. And as I started to research it more, I just found more and more adversity and more complications, and I really felt maybe this is kind of this spirit telling me, don't do this, I don't want you to do this. And I took that to heart, and so that was kind of hard for me to kind of reorient what I wanted to do. It's just more stuff. So I started thinking about what are some of the other ways that I could do psychedelic research. And so I've actually made, let me just kind of get through this. So this is kind of like the overarching purpose of my lab here at the university, is to study trauma. But I feel that trauma is sort of like this core feature that manifests into all these different disorders that we think are totally separate, like depression and PTSD and anxiety and addiction, when in fact they all kind of have this common core of some traumatic experience that then is manifesting in these different ways. And it looks like there's all these different psychedelic compounds or psychedelic-like compounds that are very helpful at treating this sort of complex constellation of traumatic symptoms. And I found that very interesting. And if you want to go to my lab website and look at, I have this network on my website, and each of these sort of red things is a list of references from the scientific literature that supports this sort of link between, like psilocybin treating addiction or anxiety or depression. So if you want to dig into that and nerd out on that, you can go there. And then these are just some other things that are in there. But the thing that I was focused on for so long was ayahuasca for PTSD. And so this line is still kind of a hash because it's only been kind of a hypothesis. It's only been proposed in a review that I've written in a book chapter, and talks that I've given, and kind of anecdotal information from these survey studies I did, but not a formal clinical trial to really test whether this is helpful. But if you get enough anecdotes saying something helps, I think it's worth investigating. So hopefully somebody else can pick up this work. But now what I'm kind of more interested in, because I am a neuroscientist, and I'm really interested in the underlying mechanisms of how these drugs work, because I think that would be really helpful to understand so that we can actually pair the appropriate therapy with the appropriate drug. And so a lot of my research is focused on kind of like precision medicine and identifying subtypes of these disorders and what treatments are most appropriate. It could be psychedelics, it could be something else, it could be your standard line of pharmacotherapy or cognitive behavioral therapy. I think it really just depends on what type of disease they have and their full medical history and whatnot. But a lot of the research into psychedelics right now is focusing on whether they have any therapeutic potential, but not so much on the mechanisms of how they work, because research on these subjects has been kind of really difficult to do given the war on drugs. So there was a lot of research into these compounds in the 60s and the 50s and 60s. Psychiatrists particularly were very interested in these compounds. But with the Controlled Substances Act, after the Vietnam War and the hippie counterculture movement and all the protests and everything, and for whatever reason in 1970 we got the Controlled Substances Act, so that made all of these drugs illegal, and it made research into them almost impossible. And so there was kind of a 30-year lull in research. We saw a slow emergence of it where people were starting to do animal research, and then the first major study that happened was in the 90s with Rick Strassman doing his famous DMT spirit molecule study, where he actually gave people psilocybin in a hospital, or not psilocybin, DMT in a hospital setting. So how many people are familiar with Rick Strassman and DMT, the spirit molecule? All right, so for those of you that don't know it, there's a documentary about it on Netflix that you can watch. It's narrated by Joe Rogan, and it features prominent scientists in the field that have been working on this, as well as Rick Strassman. And then it's also got some testimonials from people that were in the study, kind of talking about their subjective experience and how they felt about it. And so it's a really interesting film if you want to watch it, or you can read the book.
What is it called?
The DMT, the spirit molecule. There's a hypothesis out there that DMT is connected to your soul, that it comes into the fetus 49 days after gestation, which the Tibetan Book of the Dead also says that's when the soul enters the body. And then there's this theory that there's a massive amount of DMT released when you die, and that's when your soul's leaving. I don't know if that's true, but it's kind of an interesting, there is more neuroscience research coming out in animal models that's kind of being able to show where it originates in the brain. And we actually, Christopher Gunlock in our group actually did a science club about that a couple of weeks ago. Anyway, so that study, that DMT study, was kind of the first big clinical study giving a Schedule I drug to somebody after the war on drugs started. And then that slowly started to, I think, inspire more people to do research. So then we have Roland Griffiths in 2006 doing research on psilocybin, basically giving psilocybin to healthy people and seeing how it affected their mystical experiences, which is kind of a weird study. He's a pharmacologist, he studies drug abuse, and one of his colleagues, Bill Richards, who is this kind of very spiritual clinical psychologist that's been working with him at Hopkins, had noticed that Roland Griffiths was very into his meditation practice and yoga and kind of mindfulness. And he was like, hey, you know what's really good at inducing that? It's kind of like a positive control for your mystical experience is psilocybin. And so that kind of piqued his interest, and that kind of started that whole wave of research. And that had the whole field has just blossomed since then. There's been people doing it at UCLA and Harvard and NYU. It's a really exciting time to do psychedelic science. Where was I going with that? Yeah, but most of it's been focused on, does this affect some behavioral outcome, but not what is it doing in our brains? So I've become interested in psilocybin for several reasons. One, I think it's a little less stigmatized than things like LSD. It's a little easier to work with, and there are actually pharmaceutical grade versions of it that Maria Sabina, how many people know who Maria Sabina is? So she was one of the first people that gave psilocybin mushrooms to, I think, was it Gordon Wasson? Is that right? In the 50s, and that's kind of how it kind of exploded into the hippie counterculture, was them going down. And so she actually came up and took some of this synthetic psilocybin that they're using in the research, and she said this has the spirit. Like, she thought it was very similar to natural mushrooms that have psilocybin. And I don't think that people can say the same thing about kind of Pharmahuasca that's out there, where basically people are just taking DMT and an enzyme inhibitor to kind of mimic the effects, whereas this might be something similar. So I feel a little bit more comfortable that it's not kind of taking the spirit out of it, but it is still sort of the stuff being used in research that I'm going to be talking about is sort of pharmaceutical grade psilocybin, not people eating magic mushrooms in a lab. So I've been steering my research now to look at psilocybin and to look at the mechanisms of how psilocybin affects our brain. When I started working here, I found this paper called The Unifying Theory of Psychedelics, and I was really excited when I read this because I noticed that the author actually was here at the University of Minnesota. And so I figured he was a faculty member here, and this is perfect, we can collaborate and start doing psychedelic science. Well, it turns out he's actually a graduate student, and he was in search of a thesis advisor. So he's joined my lab, and we're actually working to get started on the first psilocybin study here at the University of Minnesota. We actually just finalized our protocol this week, and we're getting ready to submit it to FDA next week. I don't want to go into too much detail about it, but this is kind of what the study design looks like. We're going to start off with a small pilot study just to kind of test the feasibility of implementing this protocol. And we're going to be looking at visuals, some mechanistic visual neuroscience using a very well-established surround suppression task that's pretty well established in visual neuroscience. This isn't going to be a therapeutic trial. We're not going in patients. So, like I said, I'm a neuroscientist. I'm not a clinician. I'm really interested in the neurological mechanisms of these compounds. So we're going to be looking in healthy populations. So we'll be enrolling. The total study is going to be about 40 people, males and females, ages 25 to 60. We want to choose 25 because the brain really isn't finished developing until 25, and I just feel like that's a safer age to start. And a lot of the clinical trials right now being done at other institutions are kind of using the same criteria. We're taking any demographic group. We want people to be in good physical health, not having any major mental illnesses like psychosis or PTSD or depression or personality disorder. We want people that have experience taking psilocybin. So if you're finding that you meet this criteria, maybe keep us in mind in the next 3 months when we anticipate to get started. We want people that have experience taking psychedelics, specifically psilocybin, because it's going to be kind of a weird paradigm where you're going to be sitting in a hospital room and we're going to put an EEG on your head and we're going to ask you to interact with this computer task. So I think that would be a little weird for somebody that's never even experienced that state of consciousness. So, to start off, we'd really like people that feel comfortable being in that space, so that we're not making people too anxious or causing any adverse events. And then, was there a question?
episode good music?
Yeah, so we're gonna have, so we're basically just gonna have people sitting in the room for 8 hours, and about, we're gonna have them put on eye shades and music headphones and encourage them to go inward, and then at 2 points during the experience, we're gonna pull them out to do these psychophysics and EEG tests. Yeah, and we're gonna be recruiting people kind of within an hour of this location. So all those different counties that you see here, I don't know if I hit them all, and then the exclusion criteria, there's kind of a lot because we want to make sure we're not getting anyone with any major mental illness or anyone that's taking any contraindicated medications that don't go well with these classical psychedelics, any major physical illnesses like diabetes or epilepsy or cardiovascular disease. And then we are going to be doing resting state MRI on people before and after. And so there are some contraindications for MRI. You can't have any metal in your body. If you had a tattoo before a certain time when they used lead, that's an exclusion criteria. There's just kind of all these things to consider when you stick someone in a giant magnet. But I'm really excited about that aspect of the study because the Human Connectome Project actually lives here, and this is actually where MRI technology was invented. I didn't know that until I got here. So we have some of the most advanced neuroimaging. We have the most advanced neuroimaging facility in the world here. So I'm really excited to kind of dig in and see what kind of cool neuroimaging results we can get with psychedelics and get like a psychedelic connectome or something. All right, and all this work is supported by grassroots fundraising. I have a little bit of startup funding from the department, but it's not enough. I think the whole study is going to cost like $1.6 million. I worked out the budget today. Over a 5-year period, that's to pay for people's time, to compensate participants. The MRI is not cheap, and then paying for study staff. We want postdocs and clinical research coordinators, and so it's just this is kind of the normal cost of doing a study. It sounds like a lot of money, but this is kind of what it takes to do this kind of work safely and correctly. So we have there was a large donation made to me like last November. Someone in the community donated like $20,000 wanting to help support psychedelic research at the university, and so I wasn't really sure what to do with that amount of money, so I contacted the university, and the University of Minnesota Foundation is basically a nonprofit where they can take tax-deductible donations. So there's now this fund set up, it's called the PATH Fund. So if you know anybody that wants to donate to psychedelic research here and support this study, I encourage you to send them that way, and hopefully they can make a tax-deductible donation. I'm praying that Tim Ferriss will want to bestow me with a lot of money. I don't know who here knows who Tim Ferriss is? Yeah, so he just gave $17 million to Johns Hopkins to start the largest psychedelic research center in the world. And then he posted on Twitter, "Where else can I sprinkle some psychedelic rocket fuel?" I was just like, yeah. So we'll see. I think once we get started and we start generating data and we start putting what we're doing out there, hopefully it'll get more attention. But I've been trying to be very quiet about this and not really advertising it much until we're about ready to get going. So we're about ready to get going, so I'm starting to talk about it. Okay, so that's it for me and what I do. Let's get on to the topic of the lecture. So what is pharmacology and why is it important to understand for psychedelics? So I just went to Wikipedia for this. I love Wikipedia. I feel like it's a great resource. So it says pharmacology is the branch of biology concerned with the study of drug or medication action, where a drug can be broadly defined as any man-made, natural, or endogenous. Endogenous means something within your body. Molecule which exerts a biochemical or physiological effect on the tissue. It's the study of the interactions that occur between a living organism and chemicals that affect normal and abnormal biochemical function. So basically how drugs and different molecules affect our receptors in our brain and how that then ultimately impacts our behavior. This is kind of a funny meme I saw just when you think of a pharmacist, it's basically just an educated drug dealer or a drug dealer in a lab coat. But, it's kind of what they are, except they're kind of operating under the legal parameters of what is and isn't able to be dispensed based on the DEA's Controlled Substances Act. And then our pharmacist here is actually going to be a Schedule I drug dealer because we're going to have them taking in psilocybin, putting it in pills, and giving it to us when we ask for it. So that'll be kind of fun. But basically, it's this really very myopic and very fine-grained type of science that's really trying to explain all this complex stuff that's going on in our brain. So I've just got a few kind of examples here of, this is like, Alex Gray. I don't know how many people like Alex Gray's art. He's this amazing visual artist. He does a lot of the artwork for Tools albums and just amazing visual art that a lot of people trip to. So this is kind of like a schematic of him, of someone like dying and transcending into the next realm. And then we have kind of various aspects of how psychedelics can be used in therapy or meditation or, bonding. But then really what we're trying to focus on here is kind of the scientific investigation kind of at the molecular level, which is what pharmacology is. And here I'm showing sort of the two. So we have LSD and then serotonin. So serotonin is a major neurotransmitter in your brain, and LSD and some of the other psychedelics have very similar chemical structures to these sort of transmitters that you already have in your brain. So what do we know about the pharmacology of psychedelics, and where are the knowledge gaps for new discoveries? So kind of getting back to this diagram, what I really would love is if there was sort of like a comprehensive understanding of how all of these different drugs impact all of the different receptors in the brain, and what ultimately that contributes to our knowledge of how they affect behavior and decision making and neural processes and all that stuff. So that's kind of where I'm hoping some of my research can help. But there's a lot of these compounds out there, and there's a lot of things to consider. I'm not going to try to explain all of this, but basically these are like various different analogs of different psychedelics. We have DOI and mescaline and MDMA and LSD, salvia and psilocybin. You get the picture. But what we really know about psychedelics and the pharmacology of psychedelics has been pioneered by a guy named David Nicholls. So he's a pharmacologist that was working at Purdue for a very long time. He's now an emeritus professor there, and he's since moved on and as an adjunct faculty at the University of North Carolina. And so he's done a lot of the pioneering work on what we know about psychedelics and how they impact our brains and various serotonin receptors in our brains, and I'll get into that in a minute. I'm feeling particularly inspired by this guy. His name is John McCorvey. He actually trained with David Nicholls. He either got his PhD with him or did a postdoc with him. And he gave a talk recently at this conference in Madison, Wisconsin, and really kind of opened my eyes to the importance of pharmacology and psychedelics. I'd never really considered it. I wasn't really interested in pharmacology. I didn't think, just wasn't something that was really on my radar. And it's also kind of one of these things that's just so ridiculously complicated. When you try to dig into reading some papers on pharmacology, it's just like chemistry overload. And it's a little like, oh, I don't want to go back to this. I already took chemistry. I don't want to do this anymore. But I think it's a really important thing to consider, especially if we're trying to design very specific drugs for different specific disorders or neural processes, and trying to figure out if we can design kind of like the perfect psychedelic and how we could modify them to be kind of what we need for each individual person. So the basic pharmacology of psychedelics is that it's primarily acting on this thing called the 5-HT2A receptor. So 5-HT is basically serotonin. And there's all these different subtypes of these serotonin receptors. And so psychedelics are thought kind of broadly to be an agonist for 5-HT2A. So what that means is that they activate this receptor. And that if you give something that blocks this receptor, you can block the effects of the psychedelic. And it's known, this is a sort of a PET scan. So a PET scan is when it's a type of imaging scan where you inject somebody with a little protein or a small molecule that has some like radioisotope attached to it, crosses the blood-brain barrier, or they stick it directly into their cerebral spinal fluid, and then they can actually see where these things are binding in the brain. And so they found that the more of these serotonin receptors that bind to the psychedelics, the more intense the subjective experience was. And that's kind of what this graph shows. There's kind of this inflection point that once you get a certain amount of receptors bound, you're really having this very intense psychedelic experience. So this is from a review that Robin Carhart Harris wrote a couple years ago with David Nutt. These are some prominent researchers in the UK doing a lot of pioneering work with neuroimaging and psychedelics. And they kind of schematiced out this bipartite model of brain and serotonin function, where a lot of the modern sort of SSRIs, antidepressants that are being used, are trying to hit this 5-HT1A receptor to induce their effects, whereas psychedelics are activating 5-HT2AR. And so what are kind of the differences there in terms of what they're actually doing? So it's thought that 5-HT2A activation is sort of like passive coping, whereas 5-HT2A is active coping. I'm still kind of processing that. I'm not sure what that means, but if you guys want to sort of dig into that, I recommend reading this paper, Serotonin and Brain Function, A Tale of Two Receptors, where they're kind of trying to directly compare these two receptors and the potential therapeutic targets and different drugs that hit them. But basically, there's 3 different structural classes of hallucinogens, and I'm not including ketamine here because it sort of has a different structure and hits on different receptors, but it is kind of considered a psychedelic, but it's really more of a dissociative. If you're curious about that, I'll get into it in a little bit. But the 3 main classes are phenolactylamines. It's a hard one to say. So that includes things like mescaline, which is in like the peyote cactus and San Pedro cactus and DOI. Then there's the tryptamines, like dimethyltryptamine, DMT, and psilocybin. And then you have the lysergamines, which obviously is LSD. But they all kind of have this similar structure of kind of having this like similar thing to serotonin. So up here on the top is what serotonin looks like, just straight serotonin. This is a chemical in your brain that you have. And then you can see LSD has that same kind of backbone, but then it's got a whole lot of other stuff added to it. Same thing with mescaline, although it doesn't have this sort of part right here. It has kind of only a partial part of it. And then this one has both of these and then a little extra. Maybe it's just got this extra OH group, but it's moved. And then this whole thing right here, which who knows what difference that's having, but hopefully pharmacologists can figure that out. The next thing about it is that psychedelics are targeting G protein coupled receptors. So there's several different types of receptors in your brain that various molecules can bind to. So about 33% of them are these G protein coupled receptors. So these include these serotonergic receptors I was talking about that psychedelics bind to. And they're sort of these transmembrane proteins that have an extracellular part where there'll be this sort of part of the receptor that will bind to any neurotransmitter, and then it crosses the cell membrane, and then there's this whole other protein scaffold that's attached to it on the inside of the cell that once that receptor is bound to its kind of ligand or neurotransmitter, will then activate that intracellular part, and then it starts the whole kind of cascade of activity that'll happen in the cell.
Are you okay if we snap a pic of the slide?
Yeah. I'm also trying to record this, and then I'll share the actual slides as well. But yeah, you can go ahead and take pictures. So the GPCRs are the largest family of druggable targets. And yeah, I think that's all. So for the 3 different structural classes of hallucinogens that I mentioned before, these are kind of the different receptors that they all have some binding affinity for. So the phenolactylamines, so remember those were mescaline and DOI. So those are selective for serotonin receptors, specifically 5-HT2A and 5-HT2C. The tryptamines, they bind, they're non-selective serotonin, they bind non-selectively to serotonin receptors, so they kind of hit all of the serotonin receptors. And then LSD is kind of a dirty drug in terms of its kind of binding affinity. It's a little loose, I don't want to say slutty, but it's kind of just all over the place. It's binding to all the serotonin receptors, all the dopamine receptors, and all the adrenergic receptors, and maybe more. But the one common theme amongst all these is this 5-HT2A receptor. And like I said, if you block this 5-HT2A receptor, you can basically stop this psychedelic effect of each of these drugs. So it's kind of this interesting thing where it's necessary, but is it sufficient? Like, are there other, like, what are these other receptors doing? Are these other receptors having some aspect of anxiety, or is one of these really linked to the mystical experience, or paranoia, or I don't know, like, this hasn't really been teased out because although the pharmacology has been worked out, no one's really done a study where they try to selectively block each of these one by one and then ask the person what's different about your trip. Is there a question?
Yeah, so are you saying that with the SSRIs, they do not bind to the 5-H?
SSRIs act to create, they try to get it so that there's more serotonin available kind of in the intracellular space, so they work to help prevent serotonin from being taken back up by the presynaptic cell, but then there's more of it available to bind to these receptors on the postsynaptic cell. If that doesn't make sense, presynaptic, postsynaptic, so when neurons touch, there's one neuron sending a signal that's presynaptic, and then postsynaptic is the neuron receiving the signal, and anytime you're trying to get 2 neurons to talk to each other, they do that, and they do this by kind of spilling a bunch of chemicals in between this little space. Anyway, good. Okay. So let's see. So in terms of where 5-HT is expressed in the brain, so it seems that the 5-HT1, which seems to be the target of some of these SSRIs, is more highly expressed in the hippocampus, whereas we see these 5-HT2A receptors are more highly concentrated in the cortex. And the cortex is kind of like, our super evolved part of our brain that gives us the ability to think and reason, above kind of lower animals. Like rats don't even have they have a cortex, but it's not as kind of convoluted as ours. They don't have as much surface area. But they do also have 5-HT2A receptors, and they are primarily expressed in layer 5 pyramidal neurons in the cortex. For anybody that's a neuroanatomy person, these are kind of like the primary excitatory neurons in the cortex. And so just to kind of remind you of what kind of some of this terminology is. So we know that activation or agonists of 5-HT2A are primarily psychedelics. I was looking at this the other day, and I was actually really surprised to learn this, that really psychedelics are the only drugs out there, the only molecules that bind to 5-HT2A and activate it. Everything else will inhibit it or not bind to it. So I thought that was really interesting that they're very selective for what is the purpose of this receptor if only these kind of exogenous psychedelics and DMT potentially are binding and activating these things. And then if anything is blocking the receptor, such as antipsychotics and antidepressants, then we would call that an antagonist. And it's kind of interesting because that's kind of been the line of therapy that people have gone with, is we need to block 5-HT2A if we want to treat these major mental illnesses. So I think it really flips psychiatry on its head to say, no, we need to activate them. So it's a little, okay, so we'll get with you and then that one.
Is that possibly because we haven't found the sort of endogenous function, or we ruled it out that there isn't an endogenous sort of?
Well, I think DMT might be the endogenous agonist, right? So we all have dimethyltryptamine in our brain, but it is broken down almost immediately after it's produced, so it's almost impossible to detect its effects or measure it in people. But that's really the only one I'm familiar with, as well as serotonin.
What about some kind of co-evolutionary use for those receptors?
Yeah, I mean, it's a good question. I feel like this is kind of like a philosophical debate. We've had these conversations in some of our philosophy groups in the Psychedelic Society because they're kind of esoteric and there's no real way to test them, at least that I know of. So we'll go to that question and then I'll get you.
Okay. Yeah, it's maybe a bit of a tangent, but I studied anthropology, and I had this dilemma when kind of taking on my anthropologist element, being in maybe traditional societies or in an ayahuasca ceremony, and then going back and forth between the subjective and the objective when you're kind of trying to be scientific and analyze and objectify, and sometimes you lose a lot of information because you tried to analyze it and understand it in a certain way. And I'm wondering if you struggle with that at all, because ever since I've had those experiences, I have a really critical lens of the scientific view in terms of just missing so much and being there are literally tens or hundred thousands of years of human knowledge that science thinks it's going to in a brief amount of time suddenly discover something new that people have been working at in a different scientific method for a long time. So maybe just kind of a critical lens of why start over and what is the value of objectifying these things.
Mm-hmm. It's a good point. I mean, I'm spiritual. I've had very profound mystical experiences with psychedelics, and so I kind of have this, these two sides of my life where I'm a very, staunch scientist following the scientific method and all of the regulations about that. But then part of me is still very open-minded about some of these things that don't really have empirical evidence for them. But science is constantly disproving itself and discovering new things. So I like to think that the spiritual side of me can keep me flexible and open-minded to new discoveries and not being so bound by the scientific method. I think it's a great tool to try and falsify hypotheses and figure out and get at some semblance of truth about a topic, but I don't think it's the end-all be-all in terms of discovery of truth and knowledge. But it is a good tool. I think it's just good to not think, with anything, not to be too dogmatic about it.
Just a little question about DMT. Isn't there some evidence that the body produces DMT at birth and death or something? Can you say a little bit about that?
So I think, I don't know the data on it, I've read it. I don't know where that information came from or how it was tested or proved, but it is kind of interesting. You know, I think there's also some theory that women produce a lot of it when they're giving birth to help them kind of cope with the pain, kind of put them in a bit of an altered state. But again, I don't know how that was tested. Might have been something done in a rat or something. So it's an interesting idea. I just, you know, in terms of like kind of the philosophical question of like, why does DMT exist? It's in our brain, it's in all these plants. We're co-evolving with this molecule that's giving us these profound experiences. Like, why does it exist? Why do we have this thing? I don't know. Okay, there was you and then you.
Well, I just heard somewhere, I can't remember where that was either, but that the chemical psilocybin doesn't seem to have any other function to the plant. I mean, there are chemicals that plants produce, or it's not a plant, but you know what I mean, that are either a defense mechanism, reproductive, processing nutrients or something. And it doesn't seem to have any function, and yet it binds perfectly with a human brain.
Yeah,
Kind of
it's
odd.
interesting. So there's a new research center that opened up in Jamaica that's trying to research more of like what are mushrooms doing. So I hope that's one of their questions.
Now, is this receptor just present in the brain, or can you find it in the gut and peripheral nerve as well?
Well, I know that, so I actually get into that a little later. Oh, I thought I was already showing this cool image. This is the cool neurons that are labeled. Yeah, so some of the receptors I think have activity. 5-HT2B is in the heart and the gut as well. I don't know about 5-HT2A. I know you have them at neuromuscular junctions. So you have some serotonin receptors. I don't know which subtype it is. I'd have to dig in. Are you asking or?
Asking.
Yeah, okay, yeah, that's a good question. I'd have to look into it to see where it's all expressed. Is there another question?
Yeah, do you know when we labeled this receptor as 5-H2A, like what year that was?
Mm-mm.
Yeah.
I bet Wikipedia would know. I don't know off the top of my head.
Okay.
Oh, yeah, so actually, thank you, Chris. So a lot of what we know about kind of like antipsychotics and things were developed through actually researching psychedelics in the 50s and 60s, and that's how they kind of learned that the serotonergic system was really powerful for mental illness, specifically in schizophrenia. So I think maybe it was in the 60s or 50s. Yeah.
I think Cara Harris was talking about something about the purpose of kind of these receptor types. I don't know if this was based in research or just kind of a theory, but it was kind of theorizing that like in times of need for like adaptation, like it kind of in a person's life, if there's like major stresses and it kind of need for this adaptive quality, that these can be more activated.
Yeah.
And then I was kind of wondering, because you talked about the G protein. Yeah. Talking about the G protein coupled receptor. Isn't there something related to like long-term potentiation with G protein coupled receptors versus other receptor types that kind of has an adaptive quality to it?
That's a good question. I don't know off the top of my head, but like if it's like helping to induce like depolarization and yeah,
Right. Yeah.
or action potentials, yeah. I mean, theoretically, if that's, when activating a neuron, that's kind of what you're doing, is you're trying to propagate that electrical signal from one cell to the next.
Right.
But yeah, to get at the question of kind of what's the function of activating this receptor, so this is a summary that Robin Carhart Harris put together in one of his talks, that 5-HT or serotonin is basically increasing plasticity and environmental sensitivity, and plays a key role in brain development. So you see it highly expressed in the brain as it's developing. It sort of gets pruned back, and you actually see a loss of 5-HT2A receptors as you get older. And so it's kind of thought that maybe this is involved in kind of more cognitive inflexibility as you get older, if your brain's less able to adapt or have this kind of plasticity response. The 5-HT2A receptor functioning is key in developmental periods, like I just mentioned. What's going on here? Yeah, agonism. It helps with learning and unlearning and cognitive flexibility. And I guess, it's upregulated during stress, but like any kind of time when you're stressed, you are kind of promoting neuroplasticity so that you can handle the adverse situation. But there are kind of good and bad forms of plasticity, like pain is a type of plasticity. So I think we kind of need to be careful when we think about just kind of promoting a bunch of neuroplasticity and what could that do. Like, I don't know if that's kind of when they started realizing that this is what these compounds were doing in the 50s, and then the CIA wanted to research them and thought they could be like truth serums or some aspect of mind control. I don't think that worked out very well, but I mean, it kind of makes sense that you might be able to convince somebody of something or teach them something that might not be great, or it could be wonderful. I mean, that's the whole idea behind why they might be good for therapy, is because in order to get out of an ingrained pattern, you need to kind of unlearn it and then relearn something new, and you need your brain to be in this plasticity state in order to do that. Okay, let's see. So this is just kind of getting at some of the other areas that the 5-HT2 receptors are important for, both on and off target effects. So the 5-HT2A, which is the primary one we've been talking about, has its action in the central nervous system. So this is basically the brain and the spinal cord. And this includes things like psychedelics to activate it, and antidepressants and antipsychotics to block it. The 5-HT2C receptor is involved in cognition, anxiety, and appetite suppression. And then the 5-HT2B is, I think, expressed in heart tissue and arteries, as well as in the stomach and the gut. And I'll get to that a little later, but there was this study that found that when they first actually were able to identify the full structure of LSD bound to a serotonin receptor, it was in 5-HT2B. And it turns out that it actually locks into the receptor, and that's why there's such this long-lasting effect. But that there was potentially, like, that might be why people have kind of anxiety and increased heart rate on LSD, because it is maybe overactivating some of these receptors in the heart tissue. And that might be a potential risk for anyone with a cardiovascular disease. But I just want to point out two clinical researchers that have done a lot of pioneering work on our understanding of sort of this phenomenon that 5-HT2A really is kind of this central driver of the psychedelic experience. They did a lot of work where they basically gave this very selective antagonist to 5-HT2A called ketanserin. They basically give people this drug, and then they give them psilocybin, and they wouldn't trip. Or they'd be tripping and then give them it, and it would stop the experience. And so this has been really great, because they've actually been able to do neuroimaging studies where they give people psilocybin, and then they give people a combination of psilocybin and this blocker, and they look at how brain activity is different, and they can actually localize where these receptors are in the brain by doing this kind of study, which is actually really cool, because it's really hard to actually image kind of molecular biology in a human brain. So this is kind of a very elegant way to do that, and I think it's just really great. I've actually was trying to get this compound to add as a control group for our own research study here, to really get at some of the mechanisms of this visual surround suppression task that we're going to do, but ketanserin is a European drug, and I can't get it here. And all the other comparable serotonergic antagonists are just not safe. They cause all kinds of cardiovascular issues for some reason, and all the clinical trials that have tested them for various things have failed, and so I just didn't feel comfortable using it. But it would be great if I could find some pharmacologist that could design the perfect serotonin, 5-HT2A antagonist that doesn't have all these other side effects, that we can do some interesting mechanistic research. But for now, we basically can't for safety reasons. But kudos to them for basically, figuring out a lot of this really cool stuff about where 5-HT2A is in the brain. So just kind of getting back to this idea that we think 5-HT2A is necessary for the psychedelic experience, but is it sufficient? So what are the other receptors doing for the subjective experience? And what about all the other drugs like ketamine and MDMA and marijuana? So I was trying to, in my review of this, find a really good comprehensive summary of what we know about psychedelics and the pharmacology of psychedelics. And I actually reached out to John McCorvey, who was that pharmacologist I referenced earlier that trained with David Nicholls. And his answer was basically no, there isn't an all-encompassing resource for all of this, although one could be created in a thorough review. But the long answer is that the information exists in a lot of different studies in multiple papers, usually spread out by chemical scaffold and kind of the hypothesis that they're testing, where they're either looking at tryptamine psychedelics or phenolethylines or MDMA analogs or ketamine. And so usually the studies are very specific, so it's a little daunting to try and look through the literature and find all of these. But basically, he was saying kind of the big picture about psychedelic pharmacology is that psychedelic acts as direct agonists at aminergic receptors, these G protein coupled receptors. So this includes the serotonergic receptors, the dopamine receptors, and the adrenergic receptors. MDMA compounds act at the monoaminergic transporters. So these are basically serotonin receptor transporters, and there's norepinephrine transporters and dopamine transporters, and these basically are things that will shuttle molecules back and forth across the cell membrane rather than having it being activated and inducing an intracellular signaling cascade. And there's also vesicular transporters to increase synaptic concentrations of these neurotransmitters, like SSRIs, that are kind of making more serotonin freely available. And then ketamine acts as a non-competitive blocker of another type of receptor, which is NMDA and AMPA, which they're ligand-gated ion channels. Some propose to have active metabolites at opioid receptors, but that's still kind of controversial. There's this paper that came out last year that was saying that potentially the addictive mechanisms of ketamine is happening through some of these opioid receptors. So it was a big finding. It was all over the news, but it was kind of only done in one study. It hasn't been replicated yet, so kind of take it with a grain of salt, but it's interesting to think about in terms of like the possible mechanisms of action of ketamine. So I want to give a shout out to my undergrad, Akira, who's been doing a lot of research, trying to really fill in the gaps about what, kind of to get this comprehensive review. We kind of want to put a review paper together to really tackle all of this. And so we're kind of just thinking about this side by side. Look at all these different drugs and all the different receptors and how do they act at these different receptors. So still trying to dig through what the actual binding affinities are, like how strongly they bind. Because I was thinking like you could design the perfect experiment where you're selectively blocking certain receptors because it has the highest affinity for that. So if you give that as a control group and you compare it to another one, maybe we can kind of get at some of the differences in the subjective experiences when you block or activate some of these receptors. So this is a work in progress, obviously, but all the blue area or the green areas are when there's kind of an affinity for the receptor, meaning it does bind to it fairly strongly, and then red means there's no affinity for it, and then purple is just we couldn't find any data on it. But these are just sort of like those monoaminergic, I think that was the word, receptors. But there's a bunch of different other receptors that these things hit. And I'm not including some other things, but like wanting to look at things like iboga, which also kind of has a serotonin activating mechanism. But trying to fill in the gaps for these GABA receptors, which are largely inhibitory mechanisms, glutamate receptors, which are excitatory, the acetylcholine receptors, as well as the opioid receptors, and kind of what does this all look like? So I'm excited to get this filled out. And another aspect of trying to understand the pharmacology of psychedelics is figuring out, once you've activated the receptor, what then happens inside the cell. And so the unique pharmacological signature for each molecule is determined by how and where it binds inside the receptor pocket and which intracellular signaling cascades are initiated. So for G protein coupled receptors, there's basically two main types of signaling cascades that can be initiated depending on what part of the receptor it binds to. So the receptor is kind of this big, huge protein that kind of looks like, I don't know if I can draw it. I don't want to use my Sharpie. It's like, I guess you can kind of get a sense of it. It's kind of basically like all these little protein scaffolds that are like lined up, and then like there's like a hole in the middle, and kind of these molecules can get right inside, and then there's like these different little pockets they can bind to depending on the conformation of the structure. So if it's got an extra methyl group, it might bind in one part. If you take that off, it'll bind to another part, and then that's going to activate something different inside the cell. So it's really complicated in terms of like all the possible iterations of activity that you can see. But it's generally thought that if you activate this receptor and it goes through this sort of G protein part, then this is sort of the gas, and that will turn the cell off. That will be kind of an inhibitory response. Whereas if you can activate this beta-arrestin and recruit these beta-arrestin proteins, then this signaling cascade, oh, I'm sorry, I got that wrong. The G protein coupled receptor, the G protein part is the gas. So that activates the receptor, and the beta-arrestin blocks, or it's the brakes for the activity inside the cell. Okay. So like I said, there's lots of places for these molecules to bind, and this paper is really great, again, by John McCorvey, who just kind of was showing, if you take LSD and you just change little bits of it, it activates different parts of the receptor, and it'll have a different sort of recruitment of this beta-arrestin signaling cascade. But it's thought and it's known that this beta-arrestin signaling cascade is what is involved in the psychedelic experience. So if you get that response, then you're going to have a psychedelic experience. If the G protein is recruited, then you're not going to get it. Okay. And then just to close out, I apologize for the low resolution. I couldn't find a good version of this. I don't think it's published. But this was a screen capture I took from John McCorvey's talk, where he basically was showing So each of these is like, so the y-axis is time in minutes, from 0 to 360. And then the y-axis is basically like this, the kinetics of beta-arrestin recruitment to the cell membrane when different drugs bind. And so the top one here, so the higher it is, the more beta-arrestin it's recruiting for this sort of intense psychedelic experience that we think is responsible for that kind of intracellular cascade. So this top one here is LSD, obviously. That one's going to have the most intense. And then it goes down, you have this 25, I don't even know what that is, NBOME, some research analog. You have DOI, and then 5-HT here, and then some of these other ones that aren't hallucinogenic. So I thought that was really interesting. Sorry, I have a tickle in my throat. So how are different psychedelics or different 5-HT receptor subtypes initiating these different signaling cascades? I don't think that's really known, and that's something that's being actively worked out by John McCorvey, actually. We're actually going to start a collaboration. He's collected all this data, and he's done all this pharmacological research, but he has basically more data than he knows what to do with, and he doesn't have any machine learning experts on his team to help him analyze it, and I happen to be a health informatician as like the other half of my job, so we're going to partner up and see if I can maybe answer some of these questions with all this massive amount of data that he has. But this is sort of the key figure that came out of that paper where they were finally able to show LSD bound to a serotonin receptor, where they crystallized the receptor with LSD bound into it and kind of showing the different signaling cascades that were initiated depending on just changing one single molecule on the LSD compound. So in summary, psychedelic pharmacology is hard, at least for me. It's not something that I feel a particular affinity for. I really want to understand it, but chemistry was never really my strong suit. So I hope you were able to learn a little bit from this about psychedelic pharmacology. So basically, psychedelics don't just mimic our natural serotonin. They aren't binary on or off switches for our brains. There is a difference between functional selectivity and biased agonism, and so functional selectivity is kind of relating to this idea that, you know, if there's a specific conformation of a drug that's activating the receptor, then that's considered, then it's inducing a function, then that's a functional selectivity that you're trying to select for. I think I just said that same thing 5 times. And then each of these drugs has a unique profile on how they activate intracellular signaling cascades, and to top it off, the variety of subjective experiences for each of these makes it even more complicated, because we really don't know what kind of each of these different drugs are doing. If we change one tiny little methyl group, how does that change the actual subjective experience, and is that useful in terms of designing novel drugs and therapies moving forward? So just to close it out, I want to do a shameless plug for the Psychedelic Society of Minneapolis. So we're having an event on October 6th. It's our first public event, our first public party. We've had a lot of like science-y symposiums and all these private meetings, and this is sort of like us wanting to be like, hey, let's have a party. Let's just get together and celebrate our community and support local artists. We actually have one of our DJs in the room, Simon. He's been helping bring together all the local musicians in Minneapolis and St. Paul to come and play. And then I'm bringing in Michael Garfield, who's a very close friend of mine. He's very active in kind of the Burning Man transformational festival community. If you haven't heard of him, I suggest looking him up. He is an amazing musician, artist, and intellect. He kind of reminds me of like Terence McKenna. Like he could be our generation's Terence McKenna. You just sit and listen to him talk, and he's so smart. He has so many interesting things to say. It's just, I'm so excited that he's coming. And we're gonna start off the evening with a live podcast. So he has a podcast called Future Fossils Podcast that he does, where he's interviewed lots of cool people. And so he's gonna interview me and my graduate student Link Swanson, and we're gonna talk about our study. And then he'll do a musical performance, and then we'll have various acts throughout the night playing music and have some dancing and have some fun, and hopefully it'll just be a good opportunity to gather together and enjoy the Psychedelic Society of Minneapolis. And then another effort that we're doing that I really want to help promote is, so I was mentioning earlier that there's this group called Decriminalize Nature that was able to decriminalize psychedelic containing plants like mushrooms and peyote cacti and etc. So we're trying to do something similar here. So we've rallied together a bunch of people from the Psychedelic Society, and we're trying to, so we have 6 different committees where we're trying to really tackle this issue from multiple perspectives, like getting law enforcement involved, trying to get a sense of what is their opinion about this, how do they feel about it, what would they be comfortable with in terms of implementing or not arresting people that have psychedelics on them, getting schools involved, because at some point there needs to be an education plan in place when these things become more widely available, because like sex education, you know, teenagers are going to look it up, they're going to be curious about it, and I think we need to have a responsible plan in place to educate them about responsible uses and what is and isn't okay, just like we do with sex education. The main thing is engaging with politicians, because ultimately what the strategy is to get the Minneapolis City Council to pass a resolution to decriminalize psychedelics, which would basically pull funding from the law enforcement so that they just wouldn't put any resources into enforcing any arrests or prosecutions for people that are in possession or growing or using these different plants. So we've been doing a lot of homework on who these people are. It's 13 individuals that are on our Minneapolis City Council. They're actually super cool people. I did a lot of research on them. They're all very progressive. I feel very happy about voting for them, like, because it's just, I don't feel like it's going to be too hard to convince them. It's just making sure that it's hitting on issues that they really care about. So we have, you know, groups of people really kind of doing their homework on who these people are, what the issues are that matter to them, and then trying to approach them in a way that is something that they will feel passionate about getting involved in. We have a medical engagement community because we also want to make sure that we have clinicians on our side that can really advocate for their safe use in therapeutic settings with the right kind of set and setting and support. So getting letters of support from clinicians, having them come and talk to law enforcement, talk to the politicians with us. And then we also just have kind of advocacy and education and outreach and fundraising. And we are trying to collect testimonials from people in the community. These can either be anonymous or that you can share your name, but really we want to get a lot of anecdotes from people sharing their positive experiences with psychedelics, whether they've helped them, you know, treat addiction or depression or whatever it is that has helped you in your life. We think that that can be really powerful, you know, to really tug at people's heartstrings. You know, when you make it personal, when it's anecdotal, you know, that can be really effective in changing someone's mind, especially if you can kind of tailor it towards something that's really important for them. So Chris is actually going to be helping with that. If anybody wants to submit a testimonial.
Excellent. Our website, psychedelicnpls.com. There's a decriminalization project tab at the top. Fill out a testimonial. Be a big help. You need as many as you can.
Yeah. Because when Oakland went and they presented this in front of the city council, they actually had like 30 people stand up and give kind of their little 1-minute spiel. And I mean, it looked like people were very moved by it. And so, you know, I kind of want to follow a similar process. It was very effective. And so they're trying this again in Berkeley, and they're actually trying to do a whole Decriminalize California initiative at the statewide level. That would be amazing. What else? Yeah, I think that's it. Okay.
Do you have like a timeline in mind?
We're hoping we've set the goal to have this passed by next fall.
I'm sorry, I missed the connection between the decriminalized
And the party.
The party.
The only connection is they're both being put on by the Psychedelic Society of Minneapolis.
The party better if we decriminalize.
We have our hands in there's a lot of stuff going on. You know, we have a therapist group where we're trying to get a bunch of clinicians together that when these things are available, we could have psychedelic therapy clinics, hopefully. So the private community for the Psychedelic Society of Minneapolis has a bunch of stuff. We have several meetings a week, all with different content. Like I said, we have a philosophy group, we have a recovery group for anybody that's sort of, you know, an addict but doesn't really like the format of AA or NA. And so it's a recovery group for people where they can talk about, hey, yes, I am addicted to heroin, but psilocybin has really helped me keep off of that. But then if they go to Narcotics Anonymous, they're going to shame them for taking psilocybin. So it's sort of a place that is more conducive to people's own abstinence habits without all the shame.